So this is really related to the twofold cost of sex that I talked about yesterday, I think, or the day before, in which the idea is that if you have a population, a sexual population, in which the females are providing most of the resource for the offspring, then the sexuals would be typically each producing 2 offspring by sexual union. But suppose a female just acquires a mutation that leads her to reproduce asexually, so that she produces entirely daughters. She's producing 2 daughters, both carrying that mutation. So that mutation doubles in frequency every generation. That's where the twofold cost of sex comes from. It refers to the fact that under this sort of idealized model, an allele that switches immediately to completely asexual reproduction will double every generation. That's a big effect. So now let's think about a slightly different situation, but along the same lines. We have a hermaphroditic plant, and so we have each plant has both male and female function. It has flowers that have anthers and stigmas, ovules, and so at steady state, each of these plants, I'm not sure how to draw a plant, its roots, there we are, there's a plant. Each of these is producing 1 offspring on average by seed and 1 by pollinating another plant. Okay? So we have a situation where the plant's sexually reproducing, they're all outcrossing, and plants are producing on average 1 seed, but they're also fertilizing 1 other seed somewhere else in the population. So the question now is, suppose a dominant mutation occurs so that we have a heterozygote. Let's say these are all little a, the sexuals. Now we have a big A, little a, and this big A is a dominant allele which causes self-fertilization. Okay? So that now this plant is just fertilizing itself and producing, let's say, 1 seed. Now, there are actually different versions of this. I want you to think about the rate of increase, or rate of decrease perhaps, the rate of change in frequency of this allele, given 3 situations. One is that sulfurs, as before, just produce on average 1 seed per individual. Number 2, but this is produced by self-fertilization. 2, the sulfurs actually somehow aren't bothering producing all this pollen, which is expensive, and they maybe produce 2 seeds, right? And the third possibility, the sulfurs, as before, produce 1 seed plus 1 offspring by pollination. And this actually is a plausible kind of scenario because, well, I suppose they're all plausible. This will be the situation if suddenly the flower just closed up, okay, and no pollen was exported and all the pollen milled around inside the flower and fertilized that plant, and if there was no reallocation of resources. This would be the situation if somehow the mutation just turned off pollen production, so no energy was going into the whole business of male function, and it was somehow diverted to increasing female function, maybe not doubling, but let's say doubling for the sake of argument. And then this situation would be one where, let's say you only need 1 pollen grain to fertilize the seed, so that let's say you could have a situation where the anthers move down right close to the stigma so that they self-fertilize, but they're still producing pollen, they're still exporting it. Okay? So all 3 are perfectly plausible scenarios. What I want to know is what's going to happen. Okay? In each of these 3 cases, is this going to increase, and if so, how fast? Okay? And I should say it's not a trick question like some questions we've seen, but it's not a trivial question either. Okay? So it will take a modest amount of thought. Yep. What's the question? Okay, the question is, under each of these three scenarios, we start with one heterozygote carrying a single copy of this dominant allele that forces them to self-fertilize, okay? And then there are different scenarios about how many seeds they produce and how many other plants they pollinate. Now, just forget about random fluctuations and just take the expected outcome. What's the expected rate of increase of this allele under these three scenarios? Does it stay the same frequency? Does it go up in frequency? How fast? Okay, here, so in the basic sexual population, if the population's at steady state, then each plant is producing two offspring, one via its female function by producing a seed, and one of which it provides half the genome, if you like, and the other by pollinating someone else's seed. Right? Now, if we have self-fertilization, it could be it still just produces one seed, there's no change. But it could be that because it's not producing any pollen, it can increase its female output, and it produces two seeds. In fact, the reality will be somewhere in between, and there's quite a lot of work, botanical work, in estimating the strength of reallocation of resources from male function to female function in self-fertilizing plants. It'll be somewhere between these two, probably. So some of you are not working in groups, but maybe you can talk to yourselves, and maybe that'll help. So maybe as soon as anyone has an idea about how to solve the first part, they could tell us. Anyone, any ideas yet? Well, there's only a single locus, so we're not even necessarily talking about recombination here. Yeah. So it's not, we're not having asexual reproduction. There's still, so that's crucial. So this is a heterozygote. It arises one copy. But the self-fertilization means that the offspring are going to be a quarter to a half to a quarter of the three possible genotypes. That's right. So there's still segregation. There's still meiosis, exactly. And there kind of has to be, because you have to produce, you're still pollinating, so you have to produce haploid pollen, haploid ovule, so that fertilization produces a diploid. Yeah, that's crucial. Okay, so any suggestions for the first part? So we've already got a clue from that question just now that, okay, although we're saying that a heterozygote arises, it's going to produce in the next generation, one of those is going to produce a quarter little a, a half big A little a, and a quarter homozygotes, actually, for this new allele. So actually, we can ignore these little a because the whole population is made up of the ancestral type. What we have to track is the numbers of heterozygotes and the numbers of big A homozygotes, okay? And so what we're saying is that, say through time, at time 0, we have the number 1 of these and 0 of these, and we want to know how these change through time. Okay? Maybe give it a little bit more space for this. All right. So that's the initial situation. So the question is, what's the number going to be in the next generation, and the next generation, and the next generation? Okay, so it's like all of population genetics, it's bookkeeping. It's just keeping track of the numbers. Well, I think someone should be able to get me through at least the first generation, right? So we're producing, on average, 1 seed. We've just got the heterozygotes. What's the expected number of these two classes in the next generation? Sorry? Well, it's relative to 1 of the parent. What's it going to be in the offspring? 0.5. It's going to be a half, because there's a quarter to a half to a quarter segregation. We ignore the quarter little a, because they are identical to the rest. So we have a half of this, 1, and we have a quarter of these. So now in the next generation, 1/4, yeah, and then 1/8, right? Because a quarter of these, quarter times a half, plus, but all of these are going to produce the same thing. And now we're going to go on. The number of heterozygotes seems to be diminishing, goes to 0, and they're transmitting to here, but we can forget the input eventually of the heterozygotes, okay? And these are just going to persist, right? So, this will be 1/16 plus a quarter. So it's actually the frequency of the big A is going to stay the same. Of course, in reality, there's random fluctuation. This is a kind of expectation. So probably it'll be lost by chance. But actually, if it's a certain frequency in the population, in the long run, it'll stay steady. Okay? So what happens if we have the same situation, but now the sulfur can produce twice as much seed because it's not wasting effort on producing pollen? The heterozygotes will be lost eventually, that's right. But what's going to happen to the big A homozygotes? But will they increase? That's the thing. They'll increase. They'll increase by twofold, because what's happening is that we just double each row by 2, because in every generation. So we're now going from 0 of these, a half. This is going to be 3/8, 3/4, and so on. In the long run, it's actually going to double every generation because you're producing 2 seeds. So this is really very, very similar to the standard cost of sex argument, in which basically, once you've got over this stage of having heterozygotes and you're a homozygous sulfur, you're producing twice the reproductive output as seed, and those seeds are all genetically identical to you. So you have this doubling. Okay, so now let's see if you can handle this last one. And I think now you're going to have to really write down an expression. What we want is to say, what are the new numbers of, in the next generation of heterozygotes, given the numbers of heterozygotes and the numbers of homozygotes in the previous generation, and we want another expression for this. So I'm asking, what are those question marks, right? And we've already done half the problem because we've worked out, we're assuming here we have one seed, and so one part of it is going to be the same as the previous question. A heterozygote produces on average a half as of a heterozygote by seed production, and it'll produce on average a quarter, is that right? No, sorry. It'll produce on average, yeah, it's right down here, a quarter of a homozygote by seed production. So each heterozygote produces a quarter of a big A and a half of a big A little a. And each homozygote produces by seed production, we're going to entirely 1, isn't it? And it doesn't produce, a homozygote doesn't produce any heterozygotes, okay, via selfing, okay, via seed production. But now we've got an extra output due to pollination. So someone tell me what these extra entries are going to be in here. So we're asking, we've now got a heterozygote, and it's sending out pollen, 1/2, yeah. So it's going to produce half a heterozygote by pollinating other plants. But the other half of its pollinated offspring are going to be little a, so we ignore them, yeah. And it can't produce any homozygotes by pollinating other plants, because the other plants are all little a. Okay. And now ask, what do the homozygotes produce by pollination? They produce a whole heterozygote, yeah. Is that right? Yeah, because they'll pollinate the others. And they won't produce any homozygotes, okay? So we can write this as a sort of matrix thing. The heterozygotes can pollinate other heterozygotes, but we're looking at a situation where this new type is very rare in the population. So we're looking at the initial increase. It gets much more complicated if we have to start looking at all the combinations of everything. So this is just the initial increase problem. So we could sort of write it here as 1, 1, and a quarter, and 1. Okay. So anyone tell me, how do we work out what's the long-term rate of increase? I mean, we can work through, obviously, from one generation to the next, but what's the long-term outcome going to be? How do you work that out? sure some of you know. Sorry? Yes, but we want to know how fast. The thing is, if we work through this, let's go through an example. So we have one of these and 0 of these. And so 1 and 0, okay, goes to, we've just got the heterozygotes. Actually, have I got the rows and columns right around? Probably not. Yeah, I have, I think. Okay. That will go to, no, that's right. We've got just these. We have one heterozygote in the next generation, and we have a quarter of the homozygotes. It's the next generation. Someone tell me, what's the next generation going to be? We've still got this one heterozygote, so that's contributing 1 1/4 to the next generation. But now we've got these, and the homozygotes are going to produce 1 homozygote and 1 heterozygote. Okay, so we've got a quarter and a quarter also. We add those up. We have 1 1/4, and we have a half. Okay, so I keep doing this. So I'm going to get numbers, and they sort of change, and they sort of go up, and so on. And, you know, I could plot it on a graph like this. Numbers of big A, numbers of big A, little a. So 1 and 1. No, let's do 1 there, 1 there. So we start with just one of these. And then we go to one of these, but a quarter of the other. So we go to there. And then we go to 1 1/4 and a half. And it sort of wiggles, right? You know, what's going to go on in the long run? Because in the long run, we really just want to know, is it going to go up? And if so, at what rate in the long run? What percentage in the long run? Anyone see how to do this? How many people have done physics? There you are. You definitely should know. But there isn't an equilibrium, actually, and this is a linear problem. Either it's going to go up or it's going to go down. Or yeah, so actually this is something that John mentioned. If we call this matrix Q, then the numbers change in proportion to e to the qt, sort of. Well, not quite. No, it's a discrete time problem. Q to the power t, so we've got a power of this matrix. And you multiply the matrix Q, 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 and that tells you everything. But that's also a bit tedious. There's a simpler. Sorry? Eigenvalues, that's what I was looking for. Eigenvalues, they should be really familiar. So if you have a matrix equation, which you're going from, you know, n0 to q times n0 to q squared times n0 and so on, the long-term rate of growth is dominated by what's called the leading eigenvector, which is the component that grows fastest, and it grows at a rate given by the eigenvalue. And we calculate the eigenvalue by solving this equation, taking the determinant of that equal to 0. And in this case, if we work through that, we find that, calculate the determinant equals 0, and we find that lambda equals 3/2 or 1/2. This is the leading value, which will dominate in the long run, and it tells us that the frequency of this new type will increase by 50% per generation in the long run. That's what it comes down to. Guess who derived this result first? Fisher, yes, good. You're getting the hang of population genetics. It wasn't Darwin. Darwin didn't like maths, really. Okay, so we can just start the next one. Actually, you might get warmed up to this, and we might be able to get through it. The second one is also about the advantage of sexual reproduction, but thinking about the contrast between a sexual and asexual population in the long run. This is a short-term disadvantage of outcrossing versus selfing. Now we're going to look at the long-term advantage of a sexual population. Okay. Oh, I've got plenty of clear board here. So we just say beneficial alleles arise by mutation, and they all have a selective advantage of plus 5%. Keep it simple. And we have a total per genome mutation rate of 10 to the minus 9 per genome per generation. And we have a total, let's say, haploid population of 10 to the 9 individuals, okay? And we have, initially, we'll think about an asexual population, and we think of this Fisher-Muller type argument, t equals 0. We wait, and at some point, a new mutation arises, and it establishes. In other words, it rises to a sufficiently high frequency, it won't be lost by chance, and it starts to increase. So one copy arises, stuff happens, it gets established, okay? So the first question is, what is this time to establishment? What's the expected time before the first successful mutation, a mutation that will fix in the population? What's the time before the first successful mutation arises? And secondly, what's the time it takes to get to high frequency? Let's say 50%, just to keep it simple. And so what will happen if this is a scale of 0 to 1? Actually, the Fisher-Muller argument's a little bit misleading, because what really happens is that it diddles about at almost invisible frequency, and then it basically sweeps to high frequency like that, okay? So the first question is, what are these two times? Okay. And I've given you the parameters, so these are numerical answers, rough numerical answers. I may as well give you the full set of questions. So first, how long in expectation until the first successful mutation arises? Now, it's a little bit tricky here because it doesn't know it's going to be successful, but after the fact, we can say, what's the time until the first one that not just arises, but will increase to a reasonably large number, will escape the vagaries of random drift. And then how long until it gets from one copy through to, let's say, 50% of the population, some high frequency. And the second question I'm going to ask is, during this time, how many others establish? Okay. So the point is that, this one's, it's happy, it's increasing, but it's still at low frequency for a long time. And so other ones will arise and will establish, and they'll also get to high frequency. Or at least a reasonable number of copies, 10 copies, 100 copies, whatever. For this selection coefficient, if there are more than 20 copies, they're pretty likely to get fixed. So we want to know how many of these are going to arise during this time here. And what's the third question? What would be the expected time to what would be the rate of adaptation, okay, with sexual reproduction? Okay. And fourth, this will be faster. You can kind of guess that's going to be true. We want a numerical answer. So, final question, is the advantage of sex increasing with population size or decreasing with population size? In other words, the relative advantage of sexual versus asexual reproduction on this scenario? Is it bigger or smaller as the population size goes up? So I'll give you a few minutes to think about it, and then we can work through it. And I should say, because you'll want to carry on, I mean, the fun shouldn't stop. This is question 23.5 in the on the textbook website, and this was question 23.4, and there are lots of these. 2s equals about 10%. The numbers of mutations is n mu per generation, which is 1. So T1 is just 10 generations, right? Yep. So every generation there's a beneficial mutation arising, but only 10% of those are going to make it. Okay, so then what's the second time? How long is it going to get from one copy to, let's say, 50% in the population? So this has come up in Bruce's talk, I think, and also in mine, a sort of basic formula for selection. Okay, so T2 is 1 over S log n. Just to see where that comes from, the allele frequency ratio P over Q increases as 1 plus S over 1, the relative fitness to the power T, times P naught over Q naught. So roughly, if we're asking about the time it takes for these to be equal to each other, P to equal Q, both equal 50%, then we have to wait for e to the st times P naught to be 1. P naught is 1 over n, starts in one copy. And so we change that round. T is roughly 1 over S log n. Okay? So that's very straightforward. And for these numbers, this is going to be 20 log 10 to the 9, which is actually around 420. Okay? It's going to be 9 times the natural log of 10 times 20. So that's about 420 generations. So most of the time is taken to get from one copy up to high frequency. Okay? Just to mention that actually this argument is slightly wrong, and there's a very subtle point here. Interesting one, that if you have one copy, then the expected allele frequency changes as precisely e to the st over time, or strictly speaking 1 plus s to the power t, same, more or less the same. But the increase conditional on it fixing is actually faster, because if an allele is going to be lost, that means it's going to go to 0. Whereas if it's going to fix, actually, because the expectation of the whole process is just e to the st, the expectation conditional on making it is actually a little bit faster. And what happens is that actually, on average, the frequency is multiplied by a factor 1 over fixation probability in order to keep the expectation the same. So a slightly subtle point, but it's what's called a stochastic acceleration. And it means that actually this factor here is actually n times 2s. Okay, it takes a little bit less time than you might think, but you know, it really doesn't make a lot of difference here. Okay, so the rate of adaptation, during this time, right? We know that we've got here a total of, well, in this time, 420 generations. And we know that every 10 generations, a beneficial mutation is going to arise and escape random drift and get established. So the answer, as you might have thought, is 42, right? It's the ratio between the total number of beneficial mutations in this period, okay, which arise and establish and would fix in principle, but they're all going to be knocked out by this one. Now, of course, there's actually going to be a competition between all of these when they get high frequency, and there might be slight differences in selection between them. There'll be chance effects, but still, only one of them can win. So this 42 is actually the relative advantage of sexual reproduction, right? Because with sexual reproduction, all of these, okay, could fix. because they would recombine. And actually, with reasonably high recombination rates, they'll all establish. So there's a big ratio. So the final question, I'll just leave you to think through, is can you tell me how the relative advantage of sexual versus asexual reproduction depends on population size? So this involves generalizing from this one example, okay, to the sort of general formula. Think about that. Hmm? Hmm. It's a bit complicated. Yeah, let's think it through. So suppose you have all of them exactly the same fitness. They'll all sort of get to a high frequency, and actually they'll all persist at kind of equal frequency, jiggling with each other. But that's a very artificial situation. In reality, they'll have slightly different selection coefficients, and so the fitter one will eventually win. So actually that actually alters the outcome, because it means there is a kind of sieving process. There is a competition, a selection amongst these different ones that establish. So it's still true that only one of these mutations can win, but it'll be the better of them. And so actually the rate of advantage in the asexual population will depend on the variation in fitness between the mutants. So it does get more complicated. That's partly why I chose just equal selective coefficients, so that it's just completely random which one wins. One of them will win in the end. You know, there'll be a jostling for position, but eventually one of them is going to win, just by chance. Okay, so let's just write all this stuff out, you know, in terms of the algebra. With a sexual population, the rate of adaptation, that is the rate at which you fix a beneficial mutation, is going to be the probability of fixation of each one times the number of mutations coming into this haploid population. So that's this 2SNU we had, which was basically, where were we? Yeah, which is, you know, one beneficial mutation establishing every 10 generations, right? With asexual, actually, it's rather bizarre, but the mutation rate doesn't matter. As long as the mutation rate is high enough that there's a sufficient supply of beneficial mutations, the rate is limited by the time it takes for one of them to actually fix, because it's only after it's fixed that the next beneficial mutation will tend to arise on the good background. This is glossing over a lot of details that people have worked over, but it's a rough answer. And so if this is the limiting factor, the rate is just S over log n, or we could say 2ns if we made a slightly more refined calculation. And so the ratio of the rate with sex and the rate with asex is something like nu or 2nu log n. So it increases steeply with the population size. Okay? And that's because with a bigger population, there's a greater influx of beneficial mutations per generation, and there's more competition between them. Okay? And it's surprising, actually, this ratio doesn't depend on the selection coefficient. It depends on population size and mutation rate, the relative advantages. And so just to give a reference, this was an argument first made by this out. So this is maybe encouraging. This was an argument made first by Crow and Kimura in 1965, really quantifying the Fisher-Muller argument. And they got the maths wrong, and they lost some factors of 2 and stuff. And Maynard-Smith pointed this out in 1968, and then they made a reply, sort of embarrassed, I think. So anyway, but you know, they sort of got it more or less right in 65. It's a very simple calculation, and as I say, it's a much more complicated problem if you allow for variation in selective advantage between the alleles, and allow for the fact that actually there's a second competitive process in which there's a sifting amongst different beneficial alleles. That's it. It's lunchtime. It's been fun teaching here. Yeah. So thanks for being so lively as an audience as well. Yeah, it's been great.